Yeah, so one thing we're really interested in the lab is clonal hematopoiesis. About 10 years ago, people started appreciating that many people, as they age, develop these mutations in blood stem cells, and these stem cells can expand given the right conditions, they will expand and make up a large proportion of the blood system. And people with clonal hematopoiesis generally don't have any abnormalities in their blood, but they do have a large proportion of mutated cells. So we're using the single-cell sequencing approaches that I talked about earlier to better understand why these stem cells gain a competitive advantage. For example, we're acquiring different pieces of information from the same cell. So I already talked about gene expression, but now we can look at epigenetic states as well as mutations that are present in those individual cells, and T-cell receptor sequences, which will have implications for immunotherapies. So the way that these studies of clonal hematopoiesis tie into prevention is because often years before the onset of acute myeloid leukemia, you can see an expansion of mutated cells in the bone marrow. So if we can identify patients with these mutated cells that are at high risk of progression, maybe we can do something before leukemia develops and prevent the clonal expansion from transforming into leukemia. When leukemia presents, it's a different story. So we also have a project in the lab where we look at a new combination therapy for acute myeloid leukemia. And it's actually been really nice to see in the last years how many different combination therapies have been tried based on a better molecular understanding of the disease. So targeted therapies for FLT3 or IDH or BCL2 have all improved outcomes of AML patients. And we're also now combining some of these therapies with translation inhibition to see if we can broadly target the leukemia cells but leave the healthy cells intact so that patients can have longer survival.